Yes, students, just few minutes back, I told you about androecium. Its individual unit is called stamen, and the parts of a stamen are filament, anther, and connective. It's a tissue that connects the filament and the anther. Now let us see the details of it. Anther is a bag or a sac-like structure which in future produces pollen grains. Now this anther may be of a single lobe wherein we call it as monothecous anther. Monothecous anther. Even if it is single lobe, it has two spherical structures in it. These are called Microsporangia. Microsporangia, they are circular, okay, they are circular bag-like structure. Now on the other hand, the anther may be bilobed. In that case, we call it as Dithecous anther. Okay, if it has two lobes. Anther with single lobe is monothecous anther. Anther with two lobes is said to be Dithecous anther. I told you, if it is a single lobe too, it will have two microsporangia. So thus now you can imagine that these are the four microsporangia because it is dithecous. Each bag-like structure has two microsporangia. Finally, we find here we say two microsporangia, here we say four microsporangia, or we also call it as tetrasporangiate. Sporangiate. Okay, we call this as what? Tetrasporangiate. What does the LS of anther show? It is a slanting LS that we show here. See, it appears like this. And we find the microsporangia. Okay. Please remember, these microsporangia later at maturity join to form pollen sacs. When after the formation of pollen grains, they join to form pollen sacs. That is, in each lobe there are two. So, two of them join to form pollen sac. One pollen sac here, one pollen sac. Thereby, I said two pollen sacs. Okay. Totally, we say it is two pollen sacs. Yes, now. How does microsporangia appear and what it is? Each microsporangium, single is microsporangium, plural is microsporangia. Each microsporangium is a circular bag-like structure covered by four walls. The innermost wall is said to be tapetum. The innermost wall is said to be tapetum. It is rich in cytoplasm, or I can say it possesses dense cytoplasm and multinuclei. So it is multinucleated with dense cytoplasm. Okay. Next to this, we see 2 to 3 layers of cells. Okay. Just start from the center, mark tiny vertical lines and then join this. Okay. This is how we have to draw. Please remember to have a rough sheet, a rough book, paper, pencil, eraser, etc. Whenever I draw the diagram, try to draw it. If not, soon after I finish too, you can draw looking at the technique. Is it clear? Not listening to the lesson today and some other day, if you go to practice, you can never do that. The same day you have to practice, the same day you have to listen, and it is the same day you have to try to answer the questions that is given below. Okay. Please be ready with a PDF too, because soon after I explain, I'll just mark the important points that already I have explained, okay, that I have already explained. I'll be marking for you and telling you which are the 1 mark question, 2 marks questions, etc. You can even have a marker, that will be best, okay. Yes, this is one layer. I told you 2 to 3 layers. Let us show 2 layers. Now, followed by this, we see one layer of cell, a single layer of cell. And then, outermost, we see one layer of cells. Yes, this is said to be epidermis. The second layer, okay, the second one is said to be endothecium. Third, I told you 1 or 2 layers. These are said to be middle layers. They are not 1, I told you 2 or 3. And finally, we have the innermost layer, which is said to be tapetum. Now, what is this circular structure? It is the Microsporangium, single. It encloses number of cells. All the cells are almost uniform, thereby they are called sporogenous cells, or they are said to be microspore mother cells. Okay, what are these called? They are called sporogenous cells or microspore mother cells, or even it is said to be pollen mother cells, because in future it produces pollen grains. It is called microspore mother cells because at first it produces microspores. Okay, this is a single microsporangium. It is the section of a microsporangium. Let me show you the enlarged diagram of this TS. Okay, this one. Have a look at this here. I'm marking. How does it appear? It shows two bag-like structures, another two bag-like structures, like this. This is the TS of a Dithecous anther showing 4 microsporangia. This is of one lobe, these two, okay. These two are of one lobe, these two are of another lobe. So, this is TS of Dithecous anther. So, this is the microsporangium, okay. This is the microsporangium, and these are the wall layers. So, that is what I have shown you, one microsporangium. Is it clear? Now, what is a stamen, microsporangium and pollen grain? Androecium is the male reproductive organ of a flower, and it consists of whorl of stamens. Individual unit of androecium is said to be stamen. What are the parts of a stamen? Filament, anther, and then connective. Filament, anther, connective. This is a long stalk-like structure which attaches to the thalamus. This region is said to be proximal end of the filament, whereas opposite to that is this end which attaches to the anther. This is called distal part of the filament. Thus, we see 3 parts: filament, anther, and connective. Now this is a sectional view. Okay. Now let me explain further. Before that, just mark this. Long slender stalk-like structure is filament. Bag-like structure is anther. In future, this anther produces pollen grain. Proximal end, just now I told you, and about the distal end. It's a bag-like structure which produces pollen grains at maturity. Yes, now have a look. The anther may be single-lobed or it may be bilobed. It may have single lobe or bilobe. Even though it is single lobed, it has two circular or cylindrical structures which are called microsporangium. Thus, in a Dithecous anther, we find 1, 2, 3, 4 microsporangia. Okay. Single lobed anther is said to be monothecous anther, bilobed is said to be dithecous anther, and it is tetragonal structure showing 4 microsporangia, that is tetrasporangiate. Structure of microsporangium, this is also a very important question. A typical microsporangium appears circular and is surrounded by 4 wall layers: epidermis, endothecium, middle layers and tapetum. Okay. Epidermis is the outermost layer, that is a single layer which is protective in nature. This one, it is protective in nature. Followed by that, we see the presence of endothecium, second layer, with thick cells which helps in dehiscence of anther and also in protection. Middle layers, 1 to 3 layers of cells are present here. Okay, that is what we say middle layers. They also help in dehiscence of anther and also they are protective in function. Thus, the first 3 layers helps in protection and dehiscence of anther, that means to release the pollen grains. It provides nourishment, whereas this is the most important one, favorite question for paper setters, tapetum, the 4th layer. It is the 4th and the innermost wall layer which has dense cytoplasm and multinuclei. It provides nourishment to the developing pollen grains in the microsporangia. I'll repeat once more, tapetum. It is the innermost layer, 4th layer, and each cell has dense cytoplasm and multinuclei. So, the cells of the tapetum are multinucleated in condition. At maturity, what happens? Pollen sac is formed by the fusion of 2 microsporangia in each lobe. That means what? Each microsporangium is somewhat circular in shape. They join to form a pollen sac. Thus, we say that the cells present in the microsporangium are of similar type and hence they are homogeneous, or we say sporogenous tissue. At maturity, these two join to form 2 microsporangia. Both forms 1. On the other hand, these two forms 1. Thereby, 2 pollen sacs are formed. Okay.